Hello everyone, I am Dr. Sujata Choudhuri, Assistant Professor and Head of Zoology Department, Barakpur Rashtraguru Surendranath College. Today I am going to talk about a very important topic, that is pedigree analysis, which is a part of our genetic practical, and it is a knowledge that one should have while studying any stream of biology. So, now coming to the topic in details. So, first of all, we should know what is a pedigree. So, pedigree is basically a family tree or family chart that are used to analyze the pattern of inheritance of a particular trait through generations in a family. Say for example, there is gray hair or premature gray hair in some of the individuals in a family that are inherited in generations, but we want to know what is the basic background or causative agent behind that premature graying of hair. So, to know that, we should know some of the terminology that will come in the topic. First, pedigree. So, pedigree is a chart that shows the presence or absence of a trait within a family across generations. Genotype is the genetic makeup of an individual. Phenotype is the expressed physical characteristics of an organism. Dominant allele, that is the allele that is phenotypically expressed in heterozygous condition over another allele. Recessive allele that is only expressed in absence of dominant allele. So, if it is in the homozygous condition, then only the recessive allele will express its trait. Then autosomal trait, the trait that is located on an autosome. And sex-linked trait, the trait or the gene that is responsible for the trait is located on one of the two sex chromosomes, means either X chromosome or Y chromosomes. Now, homozygous means having two identical alleles for a particular gene, and heterozygous means having two different alleles for a particular gene. Now, talk about some hereditary problem. We know that a child bears half of their autosome from his father and half of their autosomes from his mother. But in case of sex-linked characters or the sex chromosomes, they inherit, if the child is male, he inherits the Y from the father and X chromosome from mother. In case of female, the female or daughter gets both their X chromosomes, one from father, one from mother. So, in case of inheritance of certain traits or genetic factors, there may be two types of inheritance. One is autosomal, that is the trait responsible for the characters or the character gene responsible for the trait is located either on the autosomes or it may be on sex chromosomes or Y chromosomes. If it is located on the X chromosome, it is called X-linked inheritance. If it is located on the Y chromosome, it is called Y-linked inheritance. So, the inheritance can be basically of two types, autosomal and X-linked or Y-linked. Now, during pedigree, we will come into some of the symbols that are generally used. So, the first symbol, that is a square one, denotes a male, circular one denotes a female, this shape denotes a sex undiagnosed. There are many cases where we cannot diagnose the actual sex of the individual. This is the symbol for adopted, this is pregnancy, this is the deceased or the dead individuals. The solid color denotes the affected individuals with a particular trait. half solid means the carrier for a autosomal trait. This is the carrier for X-linked trait. So, carrier are the heterozygous condition for a particular trait. This line between one male and female is the marriage line or mating line. This double line means the consanguineous marriage, means the marriage between the first line of descendant or brother sister marriage. This line shows the siblings, so they are brothers and sisters. Now, number of children can be denoted by putting the number of that particular sex or gender, that is here there are 3 male or sons while 2 daughters. Now, this condition here the marriage line is disconnected, so it is a case of divorced or separated. This is the symbol of miscarriage or stillbirth. These are the symbols for dizygotic twins, means these conditions or these children started their early life from 2 different zygotes, means they have separate eggs and separate sperm fertilized and then their life have started. So, they are unidentical twins, and this one is the monozygotic twins, means they have started their life from a single zygote. Afterwards, the blastomeres have been separated to form 2 body to give rise to these monozygotic twins. So, they are identical. These are the no offspring symbols. This arrow line shows the individual who have come to the geneticist or the pedigree analyst. So, this individual is called propositor, so proband. This line shows the 2 marriage of the same individual. Now, as I have already told that pedigree can be sex linked or autosomal. So, there are many characteristics that are linked to these particular characters. Like in sex linked condition, if mostly males have the disorder and females are the carrier, that is half shaded or maybe normal individual, the trait is sex linked. It is rare for a female to have sex linked trait. Mind it, this is the condition of sex linked recessive condition. Now, autosomal, male and females are both affected with the trait and can be of equal number, and the trait, so that trait is autosomal. Now, coming to the either the trait is dominant or recessive. If it is dominant, the trait should come in each and every generation, and if it is recessive, the trait can skip 1 or 2 generations. Again, for those traits exhibiting the dominant gene action, the affected individual should have at least 1 parent affected, and the phenotype will generally appears in every generations, and the unaffected individuals or the unaffected parents can only have unaffected offsprings, because this is the dominant character. Now, for the traits exhibiting a recessive gene, unaffected parents can have affected offsprings, because the parents can be carrier and they are not showing any defects or the characters or the trait, and the trait can skip the generations also. So, these are the characteristics of recessive trait. Now, coming to the examples, these are the 2 examples of autosomal cases. One is dominant, another is recessive. Now, you can see that in each of the generation 1 or 2 individuals are affected. So, of course, this is a dominant character. Another thing is all the affected individuals have one of their parents affected. So, of course, it is a definite case of dominant inheritance. Why autosomal? Here male and female both are affected, and this individual, that is the male, has both the autosomal recessive condition. You can see that here the male has both the autosomal recessive condition, means the male has both the the trait has skipped the generation, means in the first generation there is no trait, and also this affected individual has both parents unaffected, means they are carrier but they are not showing any trait. Now these are two examples of X-linked inheritance. One is dominant, another is recessive. Here you can see that the father who has a defective X chromosome, and that is a dominant one, has passed these characteristics to all their daughters, but the male or the son is unaffected because son carries the Y chromosome from father, but the daughters receives X chromosome from his father, and if the one X chromosome that their father is carrying is defective, they will all show the defects or the trait. So this is a X-linked dominant case. In case of X-linked recessive, you can see here are some of the carrier condition. So the carrier individual can have affected individuals or affected daughter, and usually the mother will pass their trait to the son because the son gets their X chromosome from mother, not from father. So if mother is affected or carrier by a defective X chromosome, that will be passed to her son. So all the sons are affected. You can see where the male individuals are more affected than the female individuals, and this is a typical example of X-linked recessive characters. Now there are other points how to negatively rule out the possibility of a pedigree. So in the first case we can see that this is a case of autosomal dominant, and it cannot be recessive because the affected parents could not have an affected offspring. So it must be, parents must be heterozygous of course, but it is also a dominant case because in heterozygous condition the parents are showing the effect. This is the case of autosomal recessive. Here you can see that clearly two normal parents are giving birth to a abnormal or trait or the defective daughter. So this daughter has obviously carrying the gene from both their parents who are in carrier condition. So the autosomal recessive cannot be dominant because as the unaffected parents could not have the affected offspring, so parents must be heterozygous. Now there is another case of X-linked dominant where mode of inheritance cannot be confirmed, but 100% incidence of affected daughters from an affected father is occurring. So the X-linked dominant inheritance is very much clear because father is passing his defective X chromosome and that is dominant to all his daughters. This is the case of X-linked recessive, all males are affected. So this is a very typical signature motif for X-linked recessive characteristics where high incidence of affected sons suggests that X-linked recessive inheritance is coming from mother who is giving one defective X to all his sons. So the daughters are normal and this son may have got a normal X chromosome from his mother. So mother is basically a carrier condition or heterozygous condition. Now how to analyze a pedigree? So first of all we will get this kind of pedigree in hand. First thing, we have to number each and every generations by Roman numericals like here 1, 2, 3. Though these individuals are coming in the next step, but this tier is of the same position. So this is basically third generation is of containing 6 individuals total. Now coming to the next step, each and every individuals should be numbered by Arabic numericals. So, in the first generation we will have 2 individuals, one female and one male. So, we have numbered them 1, 2. In second generation we have again numbered them from 1, 2, 3, 4, 5, 6. In third generation we have numbered the individuals 1, 2, 3, 4, 5, 6. So, like this we have to number each and every individuals so that we can call them or denote them clearly. Now, we have to describe the pedigree in details. So, this pedigree contains 3 generations with total 14 individuals. Okay. So, pedigree 1, 2, 3 generations with total 14 individuals. The pedigree contains 7 females, among which 1, 1, this one, 2, 5, this one, and 3, 5 are affected. So, they are having a dark shades or color. Among 7 males, 1, that is 2, 2 is affected. All the affected children have at least one of their parents affected. You can see here this individual who is affected has his mother, her mother affected. This son has mother affected. So, each and every individual who are affected has their one of the parents affected. Now, we have to analyze the pedigree. First of all, there is no skip of generation. So, in every generation there is 1 or 2 individuals affected. All the affected children, that is 2, 2, 2, 5 and 4, 2 have at least one of the parents affected. So, this is a definite case of dominant trait. Now, the affected male, that is 2, 2 and affected female, that is 2, 5 got the affected dominant X allele from mother who is also affected, that is 1A. So, mother is having a dominant allele who is passing this dominant allele to both of his son and daughter. Okay. So, it is a clear case of X allele condition where mother is passing the both defective X allele to son and daughter, and as this is a dominant case, both of them are expressing the disease or trait, where the child of this is not 4, 2, this will be 3, 5. 3, 5 is also having mother affected. So, here the daughter is getting an affected allele from his mother. So, the trait is X-linked dominant. Now, we have to define each and every individual with a particular genotype. So, here we have taken this type of nomenclature. If the allele is wild type or normal, it should be capital A plus. Why capital? Because this is a dominant character, that is why I have taken the capital A symbol, and plus means this is a wild type or normal condition. The affected dominant X allele or the gene on X allele is called normal A capital. Wild type allele Y allele, it is the symbol of Y allele. So, here comes the genotyping. Now, this individual who has a normal X allele and abnormal or defective X allele, that is in heterozygous condition, is having a genotype like A plus A. This father is normal, so must not have any dominant allele. So, A plus Y. This first born is normal, so must not have any dominant allele, A plus A plus. This one is male, so it must have one Y and one X, that is defective or affected, so A Y. This male is unaffected, so must be A plus Y This is outsider, that is due to marriage line she is present, so must be A plus A plus. This daughter should be A plus and A, while because she is passing, the mother is passing this A or affected X to her daughter. So, the daughter must be expressed. Now, it is also outsider and should be A plus Y. And what about the children? The children, these all children are A plus or normal, because both of their parents do not have any dominant affected alleles, so all the children are normal. But in this case, these 4, 5, 6, sorry, it should be 6, the middle one, the daughter gets the X chromosome from her mother and thus gets affected allele and should be A plus A. Now, we have to make a genotypic table where we have to put the generation names 1, 2, 3 and individuals 1, 2, 3, 4, 5, 6, and by this table we have to put all the genotypes that we have found from the pedigree and to complete the genotypic table. So, last of all we have to infer or we have to confirm that the pedigree is X-linked dominant. So, up to this we have to go and finish completing a pedigree. Now, coming to a second condition, here this pedigree contains again 3 generations, will total 14 individuals, but the pedigree contains 7 females, among which 3, that is 1, 2, 5 and 3 are affected. These are female individuals. Among the total 7 male individuals, 3, that is 2, 3, 3, 4 and 3, 5 are affected. All the affected children have at least one of their parents affected. So, now coming to the analysis of pedigree, there is no skip of generation. So, each and every generation has 1 or 2 individuals affected. So, may be dominant. All the affected children, that is these 2, 3 or 2, 5, 3, 3, 3, 4, 3, 5, they all have at least one parents affected. So, this must be the condition of dominant trait. Now, here it is a peculiar signature of an autosomal condition where male and female both are affected in more or less equal numbers. So, maybe this is an autosomal condition. Again we have to confirm it. The affected male, that is 3, 4, this one 3, 4 has unaffected mother, that is 2, 4. Okay. So, X-linked inheritance is not possible because the son gets his X chromosome from mother and Y chromosome from father. So, his father is affected, but mother is not affected. So, the mother cannot pass the defective X allele to his son. Rather, his father is affected and father can give either Y allele, but it is not a Y linked condition because many females are affected. So, father must have given the defective autosome to the son. So, this confirms that this is a condition of a autosomal dominance, but not a sex chromosome or a Y linked or X linked, none other than autosomal dominance. So, same here, we can make the genotypic table by putting the genotypes in individual boxes. Here wild type allele has been designated as A plus, affected dominant autosomal allele has been designated as A, and because this is not X linked or sex linked character, we have not inserted any Y. So, this is the autosomal gene. The first individuals who are unaffected has A plus genotype. The second individual is a carrier condition, but it is a dominant one, that is A plus A heterozygous obviously. The second generation first individual is outsider A plus A plus. The first daughter is A plus because this is a dominant character. If so carrier condition cannot be possible here. So, she is not expressing any defect. So, she is normal completely A plus. This second child, that is the male, should be A plus A, as he has inherited autosomal defective autosomal from his mother, and thus are showing the dominant trait. She is outsider, that is 2, 4. So, she should be A plus. The last child, that is the daughter, should be A plus A or heterozygous condition, as the dominant trait is showing, and the last one is the outsider, that is A plus. The third generation, the first one is totally normal A plus, second one is also normal A plus because it is a dominant trait. The trait cannot be suppressed or cannot be recessive or cannot be hidden. So, if an individual is affected like this one 3, 3, it should be a heterozygous condition, usually A plus A, and the last child is normal, thus A plus. So, this is a normal condition or normal steps where we should do our pedigree analysis. Now, coming to a third one, this is a very long pedigree with many generations. There are 5 generations with total 31 individuals. The pedigree contains 13 females, among which none is affected. So, this is a very significant finding. Among total 18 males, 7 are affected. So, very high number. All the affected individuals are male. So, this may be a case of wilding. So, why not? We should see about that. Now, coming to the analysis, there is skip of generation, like the first generation do not have any individual affected, this fourth mother father is not affected. So, there is skip of generations, and affected children have both parents unaffected. So, these children, that is 5, 3, his both parents 4, 1 and 2 are unaffected. So, this is a clear case of recessive trait. Now, coming to the maleness of the trait, that is only male candidates are affected. So, either it is Y-linked or X-linked. So, affected son must not have unaffected father if it is a Y-linked condition. So, all the affected father should pass his Y to affected children, but it is not the case, because we can see that the 2, 3 individual, this one individual is affected, but his father is unaffected. So, if the child has a Y chromosome that is defective, the father cannot pass this Y chromosome to his son. So, this is not the case. Here also, in case of 3, 2 and 3, 4, the father is unaffected. So, this is not obviously a case of Y-linked. So, it should be a case of X-linked. Why? Because you can see that all the unaffected sons have unaffected father, that is 2, 3, 3, 2, 3, 4, again 4, 5, 4, 8, again 5, 3 and 5, 6. All have, sorry, not 5, 6, 5, 3 up to that, all the affected son has unaffected father. So, if it is a X-linked trait, then only the mother who is a carrier can pass this defective X to his son, but father who has one X and one Y cannot pass either to the son. The son gets Y chromosome for his father, but it is a not Y linked condition. If father condition is X defective, it should not be reflected in the son, because father passes his X chromosome to the daughter, but not the son. But here the son gets X chromosome from his mother, and of course this is the condition of X linked recessive trait. Now coming to the genotyping, here one change I have put or inserted, that is in wild type allele we have or I have taken small a, not the capital A as the gene, as because this is a recessive case I have denoted the gene small a, plus means this is a wild type condition or normal condition, and affected recessive allele is small a only. For Y allele this symbol is used as usual. So, for the genotypic table we can see that the first one, the mother who is a carrier should be A plus A, and father will be all normal A plus Y. Why mother should be carrier? Because her son is affected and this affected allele or X has come from mother. Now generation 2, the first one should be A plus A, this is outsider, so A plus Y. This affected child, that is the male one, or the 2-3 condition, 2-3 individual should be A Y, because he is affected with one X that is defective, and if male is defective with X chromosome, it should be expressed as all the males are hemizygous for X chromosome. Now these individuals can be A plus or may be heterozygous. Then but the 5 condition, that is 2-5, should be a carrier, because downstream they have passed this defective X allele. So for this I have put all the characters or genotypes, and in many cases these genotypes cannot be confirmed. So either it should be a wild type condition A plus, or it can be a carrier condition. If there is no confirmation further, so we can take the both genotype for a doubt, so that one of them may be correct. So here in many cases we have put A plus A or A plus both genotype for a single individual. So last of all we should give a confirmation, that is the inference, and we should conclude that this pedigree is of X linked recessive trait by proposing it. Okay. So I think for today this is all I am about to say, and hope that this knowledge will help you to later analyze a pedigree. Hope for the best, and I thank you.